Good afternoon one and all. So far we have dealt with the complete meristematic tissues, permanent tissues, tissue systems and comparative analysis between dicot, monocot root, dicot, monocot stem, and we have done dicot leaf. Okay. So, we are left over with monocot leaf, comparative analysis between dicot leaf and monocot leaf, and secondary growth from plant tissues, that is from plant anatomy. So, let us look into the monocot leaf first and then we will have a comparative analysis between dicot and monocot leaf. First you see monocot leaf. So, as how you have seen the epidermis in a dicot leaf, the epidermis will be the same in dicot or monocot. So, here also it has two layered epidermis. Fine? Yeah. In between these epidermal cells, you will have large sized cells, large sized colorless cells. These large sized colorless cells are called as bulliform cells, which are unique only to the monocot leaf. Fine. So, these are what bulliform cells. Take down parallelly. And unlike dicot leaf, in dicot leaf you have seen both palisade parenchyma and spongy parenchyma, whereas in monocot leaf you will see only spongy mesophyll cells. There is no palisade parenchyma, no mesophyll differentiation is observed in monocot leaf. In monocot leaf you can see only spongy mesophyll cells. Fine, right? Yeah. So, here in this mesophyll cells you can also observe what? Bundle sheath cells. Bundle sheath cells there in the dicot leaf you can see single layered bundle sheath cells, but here you can see multi-layered bundle sheath cells. Okay. Bundle sheath cells are many layered. These bundle sheath cells are surrounding what? Vascular bundles. Vascular bundle is conjoined and closed. Okay, conjoined and closed vascular bundles. So, what are the specific features of monocot leaf? First is bulliform cells. These bulliform cells will help in maintaining the shape of the monocot leaf both in turgid and flaccid conditions. Both in turgid and flaccid conditions in monocot leaf. Both in turgid and flaccid conditions in monocot leaf, the leaf shape is maintained because of bulliform cells. Okay. So, it maintains what? Shape of the leaf. Bulliform cells will maintain shape of the leaf. Shape of leaf both in turgid and flaccid conditions. Fine, right? Now, here you see bulliform cells are how many? Many layered. Many layered bulliform cells, bundle sheath cells. Fine. And in dicot leaf you have seen the stomatal distribution is more towards the abaxial side than in the adaxial side. But here in the monocot leaf, there is equal distribution of stomata in both adaxial and apaxial epidermis. So, what is the third difference? Equal stomatal distribution. Equal stomatal distribution. Fine, right? Any doubt anywhere? And the vascular bundles will be what? Conjoint and closed. Of course, when we are dealing with physiology, we will learn in detail, but as of now, do remember that the carbon dioxide fixation, 2 carbon dioxide fixations will happen in most of the monocot leaf, one in the mesophyll cells and second in the bundle sheath cells. Mesophyll cells and bundle sheath cells here are connected by plasmodesmatal connections. Okay. Mesophyll cells and bundle sheath cells are connected by what? plasmodesmatal connections. Fine, right? Any doubts anywhere? No. So, this is the characteristic features of monocot leaf and the differences between dicot and monocot leaf. Fine. Now, we will be looking into the secondary growth. Yeah. Secondary growth, wherein here you see the basic anatomy you see. First is what? Epidermal outer layer. Yes or no? First is epidermal outer layer. Below this epidermis, we completely it is filled with what? Cortex. Below the epidermis, it is cortex. And the last layer of cortex is what? Endodermis. Endodermis is the last layer of cortex. Below the endodermis, we have stellar region. First is what? Pericycle. Okay, pericycle, and inside which you have vascular bundles like this. So, this is the rough diagram of what? Internal structure. Fine. In this vascular bundles, we have learned xylem will be close towards what? Pith, and phloem will be close towards periphery. So, secondary growth means what? And in what type of plants you will observe the secondary growth? Secondary growth is nothing but the lateral growth which takes place in the primary plant body for increase in the girth of the plant. Okay, to increase the girth of the plant, the growth which takes place in the later part of plant life is called as secondary growth. Secondary growth is observed in gymnosperms and dicotyledons or dicot plants. Secondary growth is absent in monocots. Okay, it occurs in gymnosperms and dicots because they have vascular cambium. Okay, because they have cambium, because they possess cambium. Fine. This cambium can be vascular cambium and cork cambium. Okay, two types of cambium we have. Cork cambium is present in the cortex region, vascular cambium in the stellar region. So, vascular cambium will promote stellar secondary growth. Cork cambium will promote extrastellar secondary growth. Extrastellar secondary growth. Fine. So, stellar secondary growth and extrastellar secondary growth. What happens is, inside the cortex region, few of the cells will get meristematic again. Okay, few of the cells will get meristematic again, and they form cork cambium here. Okay, few of the cells will get meristematic, and what they form? A cork cambium. Okay, and these cells will undergo arithmetic growth. What is arithmetic growth? When one cell divides into two, one turns meristematic and one will get permanent. Okay, and then when this undergoes division, one will be meristematic and the other cell is permanent. As such, this form of growth pattern is called as what? Arithmetic growth. Okay, so here arithmetic growth, what is it seen like? Cork cambium, right? Cork cambium in the cortex region will undergo the meristematic activity, wherein those cells which are formed towards the epidermis are called as cork cells, and those cells which are formed towards the endodermis are called as secondary cortex. Fine. This is the gist about extrastellar secondary growth. About this we will learn more in the coming slides. Here you see stellar secondary growth. So, in between xylem and phloem, the cambium whichever is present is called as intravesicular cambium. This cambium is what? Intravesicular cambium. And the cambium which is present in between two vascular bundles, the cambium which is present in between two vascular bundles is called as what? Intervesicular cambium. Okay, this is intravesicular cambium This is intravesicular cambium in between xylem and phloem. This is intervesicular cambium. Based on the origin, if you have to consider, intravesicular cambium is from the beginning, okay, from the beginning of primary plant body, because it is open type of vascular bundle, but functionally it gets activated in the later part of plant life, okay. So, intravesicular cambium is from the beginning, but its function is activated in the later part of plant life, okay. So, here intravesicular cambium and intervesicular cambium will form a cambial ring here, okay. This will form what? Cambial ring. This will form a cambial ring. Clear, right? Any doubt anywhere? So, you got the gist, right? So, we have secondary growth is conducted in the plants, gymnosperms and dicots, because of the cambium. We have two types of cambium, vascular cambium and cork cambium. Vascular cambium will promote stellar secondary growth, cork cambium will promote extrastellar secondary growth. So, stellar secondary growth, we have two types of vascular cambium, intravesicular cambium and intervesicular cambium. Cork cambium will form cork cells towards the epidermis and secondary cortex towards the endodermis. Clear, right? What form of growth they will conduct? What form of meristematic activity? Arithmetic growth. This will conduct arithmetic growth. Arithmetic growth. Clear, right? Next, here you see, first we will understand the vascular cambium. We have two, intravesicular cambium and intervesicular cambium. Can you see here intravesicular cambium in each vascular bundle? Look into the theory also one. This is the TS of a herbaceous dicot stem. So, the patchy or non-continuous cambium, this is not continuous, non-continuous cambium between primary xylem and primary phloem is called as intravesicular cambium. And here you see intervesicular cambium, the cells of medullary rays adjoining the intravesicular cambium. Medullary rays which are in between the vascular bundles, they are adjoining this intervesicular cambium with intravesicular cambium and they are forming a continuous cambial ring, okay. So, this will increase the secondary growth. Because of this secondary growth, what is formed ma? See clearly. Yeah. So, observe it clearly. Here in the beginning only what is present? Intravesicular cambium is present. Yes or no? Yes. Yeah. And here at the center xylem is present, okay. At the periphery phloem is present. This is clear to you, right? Now, those medullary rays which are in between these vascular bundles will adjoin this. Will adjoin what? Intervesicular cambium. This medullary rays will adjoin intravesicular cambium and this will form what? A continuous ring. Continuous cambial ring, like this. Continuous cambial ring is formed, okay. So, this will promote the secondary growth. Those cells which are formed towards the xylem, okay, we will term it as what? Secondary xylem, okay, secondary xylem in all the cells. Towards the xylem, whichever new cells are formed, what are they? Secondary xylem. And towards the phloem, whichever new cells are formed, those are what? Secondary phloem. So, now if you have to see from pith to pericycle, okay, observe carefully from pith to pericycle. Yeah, look at it. First, close to the pith, what will be present? Primary xylem will present. Because of the cambial activity, secondary xylem will be next to the primary xylem. Secondary xylem will be close to the cambial ring. Next close to the cambial ring, what is present? Secondary phloem. Then close to the pericycle, primary phloem. And at the end it is pericycle, okay. So, see here, close to the pith, what is present ma? Primary xylem. primary xylem. Next to primary xylem? Yeah. From here you see, primary xylem is close to pith. Is close to pith. Fine. Next to the primary xylem we have secondary xylem. Next to the secondary xylem we have vascular cambium. Next to the vascular cambium we have secondary phloem. Next to the secondary phloem we have primary phloem, then it is pericycle. This order is very important. So, this is what stellar secondary growth, and this will be the order. Take down quickly. Okay? Yes? Close to the pith it is primary xylem, next to that secondary xylem, then it will be cambial ring, vascular cambial ring, and then it is secondary phloem, then it will be primary phloem, and then it is pericycle. So, this is what stellar secondary growth. Stellar secondary growth. Clear? Now, shall we move to non-stellar or extrastellar secondary growth, which is conducted by cork cambium. Yeah, here you see this is extrastellar secondary growth. Extrastellar secondary growth. Look at here ma. So, few of the cork cells will form what? Cork cambium. They will turn meristematic and they will form what? Cork cambium. Cork cambium is also called as phellogen. Cork cambium is also called as what? Phellogen. Fine? Phellogen or cork cambium, one and the same. Next, those fresh cells which are formed towards the epidermis, these are what? Cork cells. Cork cells are also called as what? Phellum. Fine. And those new cells which are formed towards the endodermis, these are what? Secondary cortex. Secondary cortex is also called as what? Phelloderm. Fine. So, phellum, phellogen and phelloderm together called as what? Periderm. Fine. Together the three layers phellum, phellogen and phelloderm together called as periderm. So, extrastellar secondary growth also clear to you know? Fine. So, from epidermis to the pith region, once you have to know the order ma. Then we will look into the secondary growth in the root and stem, dicot root and dicot stem. So, look at here carefully, from pith region we will start. Close to pith, just now we have seen what is there? Secondary xylem. Next to the close to the pith we will be having primary xylem. Primary xylem, next to that is secondary xylem, and then we have vascular cambium. Next to the vascular cambium we have secondary phloem. Very good. Next to that we have primary phloem. Good. And then we have pericycle. Yes. After pericycle, the last layer, the last layer of the cortex, that is endodermis. After endodermis it is secondary cortex, next it is cork cambium and then cork cells, and then we have epidermis. Fine. So, this will be the flow from pith to the epidermis. Clear, right? Any doubt anywhere ma? No. Next. So, now we will observe the dicot root and monocot root. So, here the vascular cambium is formed during secondary growth, but then wavy cambium is formed unlike dicot stem. In dicot stem we have seen a continuous ring, right, but there is a wavy cambium that is formed in the root. That is because of the radial arrangement of vascular bundles. So, in the root if we see, we have already seen the arrangement. In the stele you observe, just a second. Yeah, here you see we have xylem tissues, okay, and phloem will be adjoining the pericycle. This will be the arrangement, yes or no? These are the xylem tissues and this is the phloem tissues. Both xylem and phloem are not on the same radius. So, in the root we will see radial type of vascular bundles. Here you observe, ma. First, what is formed in between xylem and phloem? Cambium is formed. The tissue which is present in between xylem and phloem here is called as conjunctive tissue, and from the pericycle the vascular cambium will originate like this. From the pericycle the vascular cambium will originate and a wavy cambium is formed like this in between xylem and phloem. This wavy cambium will promote the stellar secondary growth in the root. Understood clearly? So, the vascular cambium originates from the tissue located just below the phloem bundles and the portion of pericycle above the protoxylem. Okay. Further development is seen as same as in dicot stem. The secondary growth will be same as that in dicot stem, but the cambium is wavy cambium over here. Clear, right? Yeah. Next. So, this is how you will observe in the stem we have just discussed. And after this we have what is annual ring. Okay. So, annual ring is nothing but, before knowing annual ring you should have a clear idea on spring wood and atom wood. Now, you have understood secondary growth. What is secondary growth? In which plants you will see secondary growth, because of what cambiums? Vascular cambium and cork cambium. Okay. So, as such you will observe two patterns of secondary growth, stellar secondary growth and extrastellar secondary growth. This thing is clear to you. Now, spring wood and atom wood, nothing like those dicots or gymnosperms or those plants in which secondary growth is happening. They can be in different climatic regions. For example, temperate plants. Temperate plants will have variations in the climatic regions. This secondary growth is purely dependent on the climatic conditions. So, as there are fluctuations in the seasons and different climatic conditions, you cannot see an even secondary growth. Okay. Uneven secondary growth is observed. For example, during spring season, okay, during spring season, how secondary growth is formed? The secondary xylem will have maximum thick vessels and wider lumens inside it. Okay. Whereas in the autumn season whichever wood is formed, that will be having thin vessels, okay, and they will have slow thin lumens, not wider lumens. And spring wood will be lighter in weight, whereas autumn wood will thick in weight, will be in thick weight. Okay. So, look at the clear differences between spring wood and autumn wood. I hope this is clear, right? So, based on the seasons, the wood will be different, fine. The secondary growth pattern will be different. As such, we have spring wood and autumn wood. Spring wood formed in the spring season and autumn wood in the autumn season. Okay. So, spring wood is also called as early wood or first formed wood, and autumn wood is also called as what? Late wood. These are formed when nutrients are easy to access, whereas this when nutrients are scarce, that is in cold season it will be cold conditions it will be formed. Next, here xylary elements will grow quickly and they are in larger number, whereas here you can see few xylary elements. Here xylary elements, they will have wider cavities, okay. Xylary elements will have narrow vessels. Xylary elements will have narrow vessels. Here they have wider cavities. Cambium is very active, cambium is less active over here. And then, so and the transition from spring wood to autumn wood. Spring wood to autumn wood transition is fast, whereas transition from autumn wood to spring wood is slow. Fine. This is about spring wood and autumn wood. Now you see. So, two woods are formed, one in spring season and one in autumn season. So, these two woods combinedly they will form what? One annual ring. So, the band which is formed in the spring season will be light in color. Okay. The band or the growth which is formed during the spring season will be light in color. This is spring wood. Next, the band which is formed in the autumn season will be dark in color. Okay. So, one spring wood and one autumn wood. One spring wood and one autumn wood will form one annual ring. Okay. Nothing like one light band and one dark band will form what? One annual ring. Okay. When cutting a dicot tree, the tree trunk is characteristic ring like manner. Each ring actually signifies 1 year of the tree's growth. Counting all the rings, one can tell the age of the leaf, and that study is called as, or that science is called as what? Dendrochronology. Okay. So, the light and dark bands are due to different seasons or variations in seasons. Okay. So, this is about spring wood, autumn wood and annual ring and dendrochronology. And then you can see sapwood and heartwood. Okay. Sapwood will be light brown secondary xylem. So, one point I have to highlight is during the growth, secondary growth, first listen ma, during secondary growth will be more active towards inner side than in outer side. Okay. Towards inner side of the cambium, secondary growth will be more active than on the outer side. So, towards inner side what is present? Towards the inner side of vascular cambium, what is present? Secondary xylem is present. Okay. Secondary xylem. So, secondary xylem will divide more. Okay. Secondary xylem will divide more active. As such, sapwood is nothing but light brown secondary xylem which is at the peripheral ends. Heartwood is at the center which is dark brown in color. Okay. Dark brown secondary xylem. Fine. Next is sapwood is present in the peripheral region of secondary xylem, and heartwood, this gives what? Mechanical support, and it is hard and durable. It has deposits of tannins, resins, oils, gums, aromatic substances and essential oils. It is resistant to microorganisms and insects. Sapwood, these are what? Living tissues which conducts water and minerals. Heartwood, it is dead tissue. This has highly lignified walls and they do not conduct water. These are present in the young trees or plants. Sapwood, heartwood is present in the older trees. This is the difference between sapwood and heartwood, spring wood and autumn wood. Clear, right? Heartwood and sapwood are with respect to what? Secondary xylem. Spring wood and autumn wood are with respect to the seasons, the bands which are formed, the rings which are formed in seasons. Clear? Do not get confused. Next is bark. Bark is a non-technical term actually. All the layers which are formed outside the vascular cambium comes under bark only. Okay. All regions outside vascular cambium are called as what? Bark only. So, it includes what regions ma? Outside vascular cambium means secondary phloem, primary phloem, pericycle, endodermis, secondary cortex, cork cambium, and cork cells. Lastly, epidermis. Fine. All these region comes under bark only. Okay. Clear with bark? Next. So, in bark also we have two types now, early bark and late bark. Early bark is also called as soft bark, which is formed in the early seasons. Late bark is also called as hard bark, which is formed in the end of the season. Next is lenticels. So, because of the continuous activity of cork cambium, the epidermal wall will get ruptured, okay, and cork cells which are produced, first listen ma, cork cells which are produced by cork cambium, they will have a suberin cell wall, around it, it has a suberin cell wall. After completely synthesizing cork cells, still more cells which are being synthesized because of the rupture of the epidermis, those cells are called as complementary cells. Okay, what are these cells? Which are synthesized after the rupture, because of the rupture of the epidermis, still more cells which are synthesized after cork cells, those are called as what? Complementary cells. Complementary cells. This complementary cells will not have any suberin depositions. These complementary cells are what? Parenchymatous cells, because they will broke what? Epidermis. Epidermis wall layers will be broke. And the spaces which are created are called as lenticels. Lenticels will aid in what? Gaseous exchange. Lenticels will aid in gaseous exchange. Clear, right? Okay, clear about the lenticels, complementary cells and cork cambium. Fine. Next. So, that is it with today's class. With this we have finished the complete anatomy. Let me give you a quick recap. Initially with the anatomy or the internal organization of plants, we have begun with tissues, different types of tissues. We have clearly exercised all the types of meristematic tissues and all the types of permanent tissues, both simple permanent tissues and complex permanent tissues. Then we have clearly dealt with the tissue systems, where we have exercised epidermal tissue system, ground tissue system or fundamental tissue system, and vascular tissue system. Okay. After that we have learned the comparative analysis between dicot monocot root, dicot monocot stem, dicot monocot leaf. Lastly it is secondary growth. In secondary growth also, after clearly learning stellar secondary growth, extrastellar secondary growth, we have clearly observed the secondary growth of root and stem, and then we have looked into what? Springwood and autumnwood, okay, sapwood, heartwood, okay, and about bark, lastly about lenticels. Fine. With this the complete anatomy is done. In our next class we will begin with cell cycle and cell division. With that chapter 4 will be done. Okay. Yeah, thank you.